So I've just been introducing the talk and saying a few things about what I'd like to cover today. So composition, you can think of as the total amount of habitat and the proportions of amounts of different habitats relative to each other. So you might have a lot of forest and a little bit of grassland, or a lot of grassland and a little bit of forest. That's the kind of thing we're talking about when we think about composition. Configuration is used, or habitat arrangement relates to the spatial location of different areas and how they relate to each other in space. Right, so one of the central debates has been, if we're thinking about conservation in particular, is it more important that we conserve area, that we think about the total amount of habitat we've got and it doesn't matter so much where it is? Or is it more important to conserve connectivity or the interactions, interrelations between those habitats, whether they're pieces of the same habitat or different kinds of habitat that are complementary to each other? And this was captured, Dan Simbelov's original formation was termed the SLOS debate. So you might have single large or several small, so SLOS. So, and this draws on a couple of sort of long-standing theories. So the theory of island biogeography, I'm hoping most of you are familiar with. I don't spend a lot of time on it, but basically the idea that if you've got populations nearby in space, you've got a ready source of things that recolonize following some kind of disturbance, right? As well as a whole lot of theory that arose out of that about the relative importance of the timing of different colonization or extinction events and the ways that organisms move across land or seascapes in order to recolonize or build up new communities. And it's become a central theme in community ecology as people have realized that so-called assembly rules for communities are really strongly dependent on a sequence of arrival, for example. So there's been a lot of interest in island biogeography and community ecology, but that's also translated into a lot of research in spatial ecology, people thinking about using island biogeography as the basis for trying to understand ecological processes and how they happen in space. Now, not in contrast, but complementary to that are species area relationships. This is one of the longest standing and sort of most established generalities in ecology. The bigger the area you sample over, the more species you will tend to find, other things being equal. So people have used species area relationships in their generality to say the critical thing is how much area we have, and look, if we sample over a bigger area, we will get more species. Area is what you need to think about when you're thinking about conserving biodiversity. Island biogeography people have said, well, you know, perhaps that's not quite right. It matters about propagals and disturbance, and this is not a static environment. And the counter to that is that, well, perhaps it doesn't matter because those are the patterns we see on the ground. Right, one of the big questions here is nestedness. So does the bigger area contain all of the species that are found in the smaller satellite areas or not? Because there's a question of complementarity and the degree to which you're talking about an interlinked population or actually separate areas of habitat that might be different in some way. Right, and then there's been a lot of discussion around metapopulation dynamics and the ways that this might play out if you start digging deeper into the mechanisms. So the underlying question is, how do spatial relationships between locations play out in an ecological realm through different processes? You may have different extinction rates of species in patches, so there's evidence that smaller patches are more vulnerable to disturbance, for example. There's degrees to which perturbations may modify entire patches or clusters of patches versus individual patches. So you might have a cluster of small patches, and that might be either more or less resilient to change than an equivalent area in the same big patch, depending on the kind of perturbation you're talking about. These things play into recolonization. So how easy is it for organisms to get back into a habitat once a population's been lost locally? And then there's questions about the reproductive rates and the turnover rates of species, particularly in relation to the rate at which perturbations occur in a landscape. So this is obviously a big deal with corals at the moment, where the age to maturity is gradually getting closer and closer to the frequency of perturbations. So if you have more frequent perturbations than a single generation time of reproduction, then you're probably heading in a direction where that population is going to go locally extinct. Right. some more work in the kind of the late 2000s started focusing more on the matrix. So in jargony terms, you've got patches that sit in a matrix of habitat. So now in many of the case studies you work in, that'll be coral reefs that sit in an oceanic matrix. And there's questions around the permeability of that matrix. So there might be something that makes that matrix really hard to cross. It might be predation, it might be that there's a strong current, it might be unsuitable habitat. So species won't leave a local patch, even though they could in theory do that, because they're worried about dying on their way to another patch. There's some nice examples published of that. For example, you find small passerine birds in the Amazon, little perching birds that won't cross big rivers because they're scared of predation, although they could easily fly that distance, in 5 or 10 minutes or less. So not all patches make equal contributions to connectivity. There's a strong relevance, importance of where things sit in space and how they relate to other things in the same network. This is just to get you thinking spatially. So this is a metapopulation simulation. I originally built this model to simulate the spread of rusty crayfish through a stream catchment. So what you're looking here is a river divided up into grid cells, which roughly approximate the home range of an individual crayfish. The population gets introduced up here, so it's an invasive species. You can see it, the time step is the number at the top left. Population gets introduced here, and you can see it spreads through the network. You can see a little bit earlier in the simulation, so I'll wait for it here. If you watch this point here, there's a point where you can see it starts to colonize here, and then it goes extinct locally, and it doesn't colonize there until a bit later on. Right, and you can also see that the tips of this river system, so this is going upstream, right? The tips would be colonized much later than the main channel because they're harder to get to. It takes longer for the population to work its way up here in the kind of way that a population typically spreads and replicates. So, as I said, there are organisms that can't fly, don't fly. It takes time for something like a crayfish to work its way through a complex river network and get all the way to the tips, to the upper ends of those tributaries. And people tend to forget this. We tend to think, we'll think about habitat restoration, for example, create a perfect habitat, and yay, the organisms will be there. But maybe not. You might have ideal habitat for crayfish up here, but they're not going to be there because it takes time for that population to make its way up in the stepwise pattern that natural populations follow. So the North Farrig in about 2017 kind of resurfaced this whole debate, which had been simmering along in the background, did a big meta-analysis, and she questioned whether fragmentation is in fact bad. And this is like saying a very rude word to the vicar at your tea party when you're dealing with landscape ecologists. Like, this is not good, right? But she's an eminent landscape ecologist, so her arguments got taken pretty seriously. She claims that we've been mixing up scale, habitat amount, and fragmentation. And she found from a meta-analysis that 70% of documented responses to fragmentation are not statistically significant when you correct for these other factors. So laying down the gauntlet to people saying, is arrangement of habitat really that important? Well, Fletcher and a bunch of other quite eminent people wrote a good response to this in 2018. And they skew a Farrig, basically saying that she's biased her data collection by the criteria she used to select the sites, and a few other issues, right? So I tend to fall on the Fletcher side myself, also because it fits better with my kind of common sense feeling, but it's still an active debate. So the bottom line is the results from fragmentation experiments, which get more closely at mechanism than the kind of more typical pattern-focused analyses, suggest there's quite a range of counterintuitive effects that you can get. So things, when you measure where animals are in a landscape, you're looking at a set of dynamic processes. You take a snapshot of that, and over long time periods, you can actually get quite significant changes. And there's some very cool studies looking at things like overriding founder effects, hidden relaxation effects. So that would be where you have a population that's actually kind of committed to extinction locally, but it takes a while for individuals, mature individuals in that population to die. So you have the impression of a healthy population, but there's actually no recruitment into that population. Or shifts in predator-prey relationships. There's an example of the South African coast where you actually had inversion of a predator-prey relationship. So you've got lobsters eating whelks typically, and then following a storm, there was this massive increase in whelk abundance, and they actually started eating eating the lobsters. So you get all kinds of funny things going on when you've got rapid change, small areas, and shifts in habitat of these kind of magnitude. Right. So one of the ways I approach this is to think about scale, and scale is something that we assume often, but it needs to always be treated as a hypothesis. And if we're thinking about conservation in particular, what we're looking at is things that use a landscape at several different scales. I like this framework, which comes, we've published a kind of an updated version, which I've got here, but it comes originally from some work done in the Nature Conservancy in the early 2000s. So breaking down, if you think about habitat, there's some species that basically are fine in a relatively small area over most of their lifespan. Right? They don't need a massive area to persist. There's others that use the landscape more like a network. There's others that are regional species, and then you've got things like many of the migratory birds, for example, that are basically dependent on a global habitat network. Things that move, for example, from the Arctic all the way down to the South African coast and back again every year. And they need stopover points on the way. It's a global conservation problem. And that's what things like the Convention on Migratory Species are there to do, is to try to address those habitat needs. So when we think about these, a frame of reference or an approach that I've found useful in many cases is to start thinking about them as networks and to use some of the tools from social network analysis, which have also become more and more increasingly embedded in other disciplines. So this figure on the left is from a paper we did thinking about the spread of disease through various populations. So the vulnerability axis refers to contagion, the vulnerability of an individual node in the network or the entire network to some kind of contagious effect. I just wanted to present you this because it's got a nice example here of something I'll talk about a little bit later, which is a metric called betweenness centrality. So there's various ways you can look at how much does an individual node, if you think of each of these dots as being a node and the lines between them as a link, how much does each individual node contribute overall to the connectivity of the entire network? So if you've got a metapopulation of some sort that depends on having connectivity between patches, and you've got limited conservation dollars, the argument goes that you should probably focus on the patches that contribute the most to connectivity, if connectivity is important to your particular species. So the shading here shows the betweenness centrality of every node in this particular network. Betweenness centrality is calculated by taking each pair of points and then looking at what's the shortest distance that I can use traveling through the network to get from the first point to the second point. So for example, to go from here all the way up to here, my shortest path is going to go somewhere here, and it'll take me through probably one of these nodes in blue, which are the ones with the highest betweenness centrality. And the same over here, and the same over here. Whereas these two, to get between these two, there's just this one path here, right? But these nodes don't have high betweenness centrality because those two are irrelevant if I'm traveling from pretty much anywhere else in the network to somewhere else in the network. So betweenness centrality gives you a measure of the node's individual contribution to the network as a whole. Right, so here's an example of a centrality metric. It's not actually betweenness centrality, but it's pretty similar. This shows you the Western Cape. So we're talking about South Africa here. This is the Western Cape province. It's a little smaller than Queensland, but it's a pretty big area. And these areas in grey here represent different protected areas. There's several different kinds of protected area in the Western Cape, national parks, provincial parks, and there's some private areas as well. And that's a depiction of where they fall within the Cape. To understand the geography of this, you need to also realize that there's a kind of a horseshoe of mountains around, right? So many of the areas that are conserved are high-lying mountain catchment areas. If we map out between each of those different protected areas, we treat them as a node, and then we treat it as a link that just any pair of these, we look at how far it is from here to here, for example. We might say that can be thought of as a link or potential link. Then we run into a question which brings the issue of scale to the front. What do we consider connected and what's not connected? So obviously two areas that are right close to each other in space are very likely. It's very likely that animals or plants or whatever it is can move between them. But if they're really far away, the chances of an animal getting through a human-dominated landscape are pretty slim, right? So there's a distance-related effect there in terms of how we think about or try to define that network of sites. And this shows you what this might look like. So this is, it's similar to that figure I showed you a moment ago, showing centrality of each of the nodes. And the dots in green here, hopefully you can see, indicate which nodes have higher centrality. So the bigger and darker green nodes are the ones in this network that have highest centrality. The main point here I want you to notice, so this is 5, 10, 15 kilometers is what the numbers are, all the way up to 50 kilometers. And the main points to notice are at smaller distances, everything's a bit less interconnected. That's what you'd expect. So if you say nothing is connected if it's further than 5 kilometers away, or if they're further than 5 kilometers apart, they're not connected at all, you end up with relatively sparse connectivity. Right? By the time you get up to 50 kilometers, most things are within 50 k's of each other, or many things in this particular landscape. And so the network ends up with many more links. So now if you think about this as an ecologist, well, you might have data on, let's say you've been working on a small passerine bird and you know that it can fly up to 40 k's away from its home area. So you might say, okay, this is a good distance. I'm going to work with this as my network. But what about all these other distances? What if your bird is actually a 45 kilometer dispersal capability kind of a species? Or what if you're also interested, as most protected area managers are, in thinking not just about that particular bird, but let's say we're interested in tortoises, or we're interested in some lizards that we know probably move 5 kilometers or less. How do we get a cohesive answer to which areas are contributing the most to overall connectivity as opposed to just for a single species? So this shows you what we did was consider several different spatial scales. So these on this axis here corresponds to those figures I just showed you, the different networks from 5 up to 50 kilometers. And these are the 10 conservation areas in the Western Cape that emerged as being the most important for network connectivity overall. What you need, the main take-home point here is to note that it's not consistent across scale. So some of them, like this Hexburg Nature Reserve, that's along a river and over some mountains, and that's pretty much a really important area for connectivity the whole way, right? It's arguably the most important single area for network connectivity across all scales. So that's great. That's a really clear conservation priority. That's really important. But what happens when you get down here? You can see some of these lines crossing over. So if you're trying to think about how you conserve something that moves at these 20, 25 kilometer scales, you've got a bit of a difference in priorities potentially. So there's a scale dependence to these kinds of decisions. Most conservation plans are designed at the single scale, and they often ignore these kinds of outcomes. So there's some very practical relevance to this for thinking about how you do conservation. The different species use the landscape at different scales. And how can we measure connectivity across multiple scales? And if we could, is it going to be useful or not? All right, so I'd like to introduce you to an approach that we've developed to try to solve this problem for how we measure cross-scale connectivity. So there's various kinds of centrality measure. I don't want to get too bogged down in the details of exactly how you measure centrality, node centrality, because there's 4 different ways we've tried, and we don't see huge difference between the different approaches. So degree centrality measures in network analysis are used to measure and compare connectivity between different networks, as I've kind of explained already. So you can use that node centrality to look at the contribution of nodes to overall connectivity of the network. And that node connectivity is scale dependent. So ecologists often try and address this by using dispersal data. We try and get dispersal data from 4, 5, 10 species and say, what does that tell us about connectivity? And we use that known dispersal ability to try to map out what's connected and what's not connected. Right? But that can be really challenging, and it's always only a small subset of possible organisms that you can work with, because many of them are too small, too big, too fierce, whatever the case is. And just really hard to work with, particularly in the marine environment, where it's really hard to get good tracking data. But at the same time, we know protected area networks are expected to conserve multiple species. So this multi-scale connectivity is relevant to lots of different kinds of problem, both ecological and conservation oriented. So what we've proposed in this recently published article is basically that what we can do is approach this in a more structured way. If we just think about minimum and maximum distances between nodes, this again shows you a range of different distances and how the network appears at those different assumptions about what's connected and what's not at different distances. So if we measure node centrality for all nodes using the most appropriate metric at a given relatively small dispersal distance, we repeatedly measure that up to much larger dispersal distances. And then we essentially integrate under that curve that we get. So if you think of that figure that I showed you of the different protected areas, probably the best one there, if you had to pick one as being the best, the highest contributor to network connectivity, the one you're going to pick is the one with the greatest area under the curve. So by summing all of those different distances, you're basically differencing, you're obtaining an integral under that curve, right? You could also do it by plotting out the curve and integrating more formally, but I'm not sure that's really adding a lot to the simpler approach of just summing up the different distances. And what this gives you is a single cross-scale measure. It's not vulnerable to a choice of scale, right? You basically in Bayesian stats, you call it integrating over a nuisance variable, right? You're basically covering a full range of options and saying, overall, if we assume that organisms use the landscape at a whole lot of different scales, then this measure would be an effective measure of capturing that. Right, so that's a kind of like a hypothesis, right? A starting point, but does it actually work? So the first step we did was to take a metapopulation model. If you start up there where that green arrow is, this just is a workflow that the details don't matter too much here, but we simulated patches in a landscape, in a computing environment. Combined them into a network based on a single dispersal distance, ran a full-on metapopulation simulation, so simulating a typical process of population increase, dispersal, mortality, with reasonably standardized parameters. And then save the occupancy for every patch after 200 iterations of that metapopulation model. So what we're doing is simulating a metapopulation, running it under particular conditions, saving the outcome, and then looping through that. So this loop 1 and loop 2 here. First trying that with different dispersal distances, a bit like the figures I've already showed you, and then creating an entirely new landscape, right? So the landscapes are created simply by placing patches randomly in a simulated area. And so it's pretty quick and easy. So the model, and then so for every simulated landscape, we calculated these cross-scale centrality measures. And we also summed the patch occupancy of the metapopulation across all the simulations. So what we're doing essentially is, on the one hand, we've got our measure of cross-scale landscape structure, centrality, connectivity. And on the other hand, we've got a simulated outcome based on reasonable assumptions about how a metapopulation might work, for which of those patches actually played the biggest role or was most consistently occupied in metapopulation dynamics. Right, and then we compare them. We say, does the cross-scale centrality correlate significantly with overall patch occupancy? So to cut a long story short, yes, it does. And what's nice about that is it means we've got some mechanism underlying why it matters, how it matters, and we can use it. We can then build with it. So in the particular the paper, we then compared it to some real-world examples and explored how it relates to conservation planning. Okay, I don't want to go into those details right now, but the point is it functions as a proof of concept. So this just shows you an example. This is what a randomly generated network of patches looks like in the program. There's some quite nice software in R that makes this pretty easy to do. So each of these dots is a patch of a given size, and you can see that the connections between them are links. And in this case, there's several different patches that are not interconnected with each other, right? So we've got a larger metapopulation in the middle, and then there's some other connections around the periphery. So simulating metapopulation growth and spread, and then test whether those centrality metrics predict overall occupancy of individual nodes. And this is the kind of the slide. So these show you the 4 different kinds of centrality measure. We looked at degree centrality, eigenvalue centrality, closeness centrality, and betweenness centrality. Betweenness is the one I showed you that figure of. Degree centrality is a much simpler metric. It basically just looks at numbers of neighbors within a particular distance. And on that axis there is the correlation coefficient. That's the correlation between metapopulation occupancy of an individual patch in that landscape, and the cross-scale centrality metric. And it's a significant result. So all of these were highly significant statistically, suggesting that we are indeed onto something here where the cross-scale centrality, obviously a much quicker and easier way than trying to run an entire metapopulation under questionable assumptions, for understanding which patches are going to contribute most, assuming a metapopulation type dynamic is operating in your landscape. All right, so this provides a potentially useful way of evaluating nodal contributions to network activity. Now comes the bit everybody's been waiting for, where I actually talk a little bit about fish. So an application to the fish communities of the GBR. This is the point at which the battery appears to have died, and the point, there we go. Did it skip one, or was this the?
Yeah.
Let's see if I can go back. Okay. I'm not sure how many times I pushed it. Whoops, okay. Beginning to see slides in advance now. Okay. Okay, here we go. So it stopped changing. All right, this is probably where the problem was. This is a fairly big slide. So for those of you who are not, so there should be some methods. Okay. For those of you who are not familiar with the AIMS fish data set, that's what I've used for this analysis. So it's roughly 50 different reefs sampled over a roughly 20-year time period. I think a bit longer actually. It's more like 50. This shows you the GBR. These are the different reefs, and the black points are the locations that have been sampled at. I've got a feeling some of them are not showing up. Anyhow, it gives you an idea of the scale.
So we can press play here.
Is there a play thing?
No.
Right, that's fine. It's just a large map, so it's here maybe struggling to get it through the processing. All right, so the data we used for this come from several different scales. So when AIMS collects the data, they sample the fish and they also do coral transits, and that's very fine-grained data from my point of view. Right? So that's data that goes with the fish samples at a very fine scale, and I haven't used the AIMS coral data at all. Controversially, perhaps, but to me this is very, very fine scale, and I'm not interested for this particular analysis in very, very fine scale mechanisms and influences. There's at a broader scale, within the kind of broader extent of each reef, there's a set of coral dominated habitats measured quite coarsely. So things like the proximity of reef flat, the amount of coral, the amount of sand, the amount of rock that's there at a slightly broader scale. Right? So I've measured that using the Allen Coral Atlas, which, keep in mind, is just a single snapshot, right? It's not a detailed time series like the AIMS data themselves are. At a broader reef extent, you've got proportions of different benthic habitats, so things like sand, rock, coral. And I've used the GBR features database, which is simply a rough polygon outline around the boundary of each reef, to measure what's in there. What are the proportions of different habitat at that slightly broader scale? And then there's a regional context within which each reef sits. So I've got at this, there's two things that I think are really important here. One is where the reef sits on big environmental gradients. So we've got north-south gradient and then an east-west roughly gradient between the coast and the continental shelf. I've had some lengthy discussions with David Bellwood about this. Anyway, so we've settled on, because David's a co-author on this. I have had some fishy person holding my hand through this. Can all look a bit less nervous about where this is going. So I've used the bounding box. The top left and the bottom right corners give you coordinates that define a location in space. The difference, the distance to the coast, the distance to the edge of the continental shelf, and the ratio of those relative to the entire gap from the coast to the shelf. Right, and then the network measures that I've been talking about, right? So the cross-scale centrality measures, calculated for every reef in the GBR, and then I've just extracted the AIMS data. So the connectivity doesn't only refer to the sample reefs, it refers to every reef. All right, so there's three different scales of habitat data that I'm using in this analysis. And I've excluded the fine-scale coral community data. But the other thing with this is you can't use it for prediction very easily because it's collected only on a subset of reefs, whereas these bigger, coarser data sets you can use across the entire GBR. So ultimately, somewhere I'd like to take this is to think more about predicting impacts of habitat change. And you can't do that with very fine-scale data. Right, so, and a set of consistent measures there. Right, so this gives you an example of what the Allen Coral Atlas data looked like for heron, which many of you are probably familiar with, the heron reef. It's a 5 by 5 meter pixel, so that's the scale at which things. And the other big deal with this data set is that coral and coralline algae are not distinguished because they've got very similar reflectance spectrum. So it's quite a coarse data set, and you'd actually be surprised if it related very heavily to fish occurrences at all, right? Well, this is probably not your best way to go about predicting which fish should go where. So the methods extracted the atlas data into the reef boundary data, calculated areas of different kinds of habitats, estimated different bounding boxes and centrality measures, included the GBR zone data as a rough measure of human influence. So that's based on the zonation map, which influences management. And then the main statistical approach I've used is variance partitioning. So treating fish community data as a response and looking at the other variables as predictors. The advantage of doing this over looking at species richness, which people do more commonly, is that when you do species richness, you lose a huge amount of information because you collapse everything, all that richness of abundances and things into a single column of data. So you lose a lot of information by doing it that way. It's much more effective in my view to work with entire matrices of community composition. Something to note also is that time, I included time in this analysis, the year of sampling, but it did not emerge as important. And that's not that big a deal given that most of the other data sets are quite stationary, right? It's not that surprising. All right, so what did I find? So these plots that I'm going to show you are the results of variance partitioning. And I've got two different measures of centrality here. The degree centrality actually performed the best in this instance, and the betweenness and the worst. So these are broken down. The Venn diagram shows you the total variance that's explained by the model, right? So the residuals here is 0.48. So this initial model explains 52% of the variance in the fish community, right? So that's a relatively high proportion given the kind of input data. So this circle on the left labeled local explains the proportion of variance that's explained by the local variables, the on-site variables. And the one on the right captures the proportion of variance that's explained by the regional variables. That's location on gradients, and connectivity of the node within the network, where the node in this case is a reef. So there's about 20% of variance in fish community composition that can be explained purely by regional influences, right? About 10% that's purely local. I think that would go up if one worked with, for example, the AIMS, the fine scale coral data probably explain a fair size proportion of that local. And then this is the interaction between the two, the interaction between local and regional. So it's jointly explained as a covariance outcome. So the overall take home from this is that 42% of the variance in fish community composition is in some way dependent on regional factors, such as where you sit on a gradient or the connectivity between different reefs. And that's consistent across different centrality metrics. It's not simply an artifact of the kind of measurement I'm using. So then I wanted to separate out zonation, which is treated in the previous slide as another local influence. I wanted to separate that out and say, can we pick up a distinct impact of zonation on fish community composition? So this is now local, regional, and zone. It's exactly the same data. The only difference is that I've taken out the data on zones, the management zones, and put them in another matrix. And so we've now got 3 different matrices instead of 2 in this analysis. And overall, although it only explains 3% on its own, there's quite a lot of interaction effects with local and regional variables, suggesting that management can explain up to about 11% of the variance in those fish communities. Okay, so this low direct significance means that it's going to be easy to underestimate its significance you might be looking at fine scale data in the field and not finding significant impact of management, but many of the impacts are sitting up here. So for example, the interaction between the zones and the regions. So I would think of that as meaning, for example, you might have degraded coral in a particular reef, and the management zone coral is perhaps a bit healthier, and a green zone or an area that's not being exploited or not being fished as heavily. You might have a slightly healthier community there, and that's factoring into regional patterns of dispersal. So as you start to degrade or lose individuals, lose abundance, or lose species within a particular management zone, you don't only have a local effect, you have a regional effect where recruitment from that reef to other reefs is going to be impacted. And that's why I think this large effect here, relatively 6%, it's not massive, but it's still something, is kind of interesting. So I think what it's showing is that if you're thinking about the significance of management zones for fish communities, you need to also think a bit more about regional effects. And could there be a regional sort of knock-on effect that's impacting your findings? Because the zones may actually be a bit more significant than we think. And I know there are studies that show the zones matter quite a lot for local fish abundance. But a lot of that work I don't think is really connected to the regional and the local, the different scales of the problem, right? So I think there's a clear niche there where more research needs to be done. All right, so here I've separated out the cross-scale centrality. So it's again the same data. I've just rearranged the data in a slightly different way between, again, 3 different matrices. The zones are now back into the local matrix. And I've separated out the cross-scale centrality data from the regional data that describe where you fall relative to the continental shelf or on a latitudinal gradient. So these are 2 different regional matrices now with the same original local matrix. No other changes in the data. And this is trying to separate out how much is it connectivity versus where you fall on a gradient. And you can see the contributions are roughly equal, right? So it suggests that connectivity it's not the only thing playing in here. There's location on a gradient is important, but it's actually about the same importance, which surprised both me and David when we were looking at these results, because most people, I think, would say that the connectivity is probably much less important than where you sit relative to some environmental gradient. Right, so this analysis raises quite a few questions. I think it provides pretty clear evidence that at some level, regional connectivity is important for fish communities. And there's lots of possible mechanisms why this might be so, but this is a statistical analysis. It doesn't really define the mechanisms. So I think there's a question there around exactly why does cross-scale centrality do so well in this example? What is the driver? We assume it's something to do with larval dispersal, but obviously there's all kinds of complexity in the reef, different currents, different barriers to dispersal in different ways. It's a bit surprising in a sense that we get such a clear result. Efforts to restore reefs often seem to assume that if you were to restore a reef somehow, that the fish would simply be there again. And I think this shows that that's not going to happen, right? You've got to have connectivity in the right kind of way if you want to restore a fish community as well as a reef, as well as the coral. So what it suggests is that the whole reef restoration agenda, if it manages to make progress in actually restoring reefs, is going to have to cross the same bridge that terrestrial studies have already done, which is to think very carefully about where are you doing restoration, what's the relationship of that site relative to a broader network, a functional network of other locations. Then there's a question of management and potential barriers to dispersal. So if we're saying broad-scale regional connectivity is important, I think there's a lot of practical management actions there and thinking about sediment, shipping, construction, pollution, other things that might function as barriers to dispersal, where management effort could be quite usefully expended, right? I'm not aware of much of this going on in the GBR in terms of thinking about connectivity and where are the barriers to connectivity that could be resolved. So for example, I don't know what impact it has having a major shipping channel going through part of the reef. You know, perhaps in some way that actually functions as a barrier. There's lots of work in terrestrial systems where you've got roads or other impacts of infrastructure, traffic, noise, all these kinds of things that reduce the movements of animals across the landscape. And that's been quite well shown in some cases. I personally don't see why marine systems should be any different. Something might be interested in moving across an area, but it can't because there's some kind of barrier there, whether it's a definite kind of blockage or something more to do with cues that make it turn back to where it came from. There's interesting questions about zonation as well. I'm not sure whether the GBR management plan will adapt or respond to loss of coral. If we see reduced coral cover, how will the zonation plan change? Does it have that flexibility to do any kind of response to that? And what this analysis suggests is that might be quite important. If the zones are making a difference, at the moment, the total direct and indirect effect is about 11%, but perhaps if you've got less coral, those areas that are being specially looked after might become more important. So perhaps that importance goes up to 20 or 30%. So perhaps those areas need to be expanded into the future to try to maintain connectivity between critical areas without having overfishing and knockback efforts to restore degraded habitat. And we don't know at the moment, I don't think, if the GBR is becoming more or less fragmented. It's really hard to tell. All right, so bigger picture conclusions. I think the new cross-scale centrality measure is useful. And I was surprised that we found what I think of as such strong relationships relative to some of the case studies I've looked at. Communities of animals are using landscapes at multiple scales, and we need to move beyond just metapopulation ecology to think much more deeply about the role of scale and space in some of the problems that we're dealing with. Answers may change with scale. So you might find something, there's this recent paper, I've forgotten the first author's name, Graeme Cumming is on it, as well as Science Advances, looking at, they're basically using the same data set, and they were able to predict 70% of fish community variance, but using local only variables. And this is actually typical. There's plenty of other examples of this kind of thing in landscape ecology where you get a fine scale result and you don't understand how conditioned it is on particular data sets and relationships within that data set. You start zooming out a bit and you look at broader scales and you realize that the picture might be quite different. And so lastly, I think there's a strong need here to understand some of the mechanisms by which reef network fragmentation might occur, and also to understand if it's a looming problem or not. So that's it from me. Thank you, and I'm happy to take any questions.